Hello, my name is Massimiliano Di Pietro and I'm Senior Clinical Investigator Scientist at the MRC Cancer Unit at the University of Cambridge and Honorary Gastroenterology Consultant at Cambridge University Hospital. In this short video, I would like to tell you briefly about the work that has just been accepted for publication in Endoscopy journal, on which I am lead co-author together with Dr. André Neves, working at CIUK Cambridge Institute. The article is entitled Detection of Early Neoplasia in Barrett's Esophagus using lectin-based near-infrared imaging, an ex vivo study on human tissue. This work resulted from a multidisciplinary collaboration between clinicians, physicists and biochemists working at the University of Cambridge, UK. As you know, Barrett's esophagus is a premalignant condition to esophageal adenocarcinoma, and patients with Barrett's esophagus are normally surveyed endoscopically for the early detection of dysplasia. However, early neoplasia in Barrett's esophagus is difficult to see endoscopically because conventional white light endoscopy has normally poor contrast for early neoplasia. The aim of the work was to investigate new ways of detecting dysplasia during endoscopic surveillance for Barrett's esophagus. In this work, we tested wheat germ agglutinin, or WGA, as a molecular probe to guide tissue sampling. WGA is present in edible food like pasta and bread, and therefore is not toxic. We knew from previous work done by our laboratory that WGA has differential affinity for dysplastic versus non-dysplastic Barrett's, and in particular it loses affinity for neoplastic cells in Barrett's esophagus. WGA was conjugated with a near-infrared fluorophore, as the human body has no background fluorescence in the near-infrared range. We sprayed the fluorescent WGA on the mucosal surface of endoscopic mucosal resection specimens obtained from patients with Barrett's esophagus-related neoplasia undergoing endoscopic treatment. And we used a clinical-grade high-resolution near-infrared camera to detect the fluorescent signal. The results were very interesting in that we found a significant correlation between the fluorescent signal and the degree of dysplasia, with 40% reduction in the mean fluorescent intensity in areas of neoplasia compared to non-neoplastic Barrett's. When we targeted punch biopsies on the endoscopic resection specimens using the fluorescent signal, we could guide tissue sampling towards the area of neoplastic change. These results are very important as they show that it is possible to use molecular dyes during endoscopy to guide tissue sampling in a more specific way than conventional dyes used routinely in endoscopy like indigo carmine or acetic acid. We are now working on the development of a near-infrared endoscope to translate this novel diagnostic algorithm in vivo. This could revolutionize the field of endoscopic surveillance of precancerous GI diseases like Barrett's esophagus, atrophic gastritis, or inflammatory bowel disease. I hope you will enjoy reading this paper, and thank you very much for your attention.